Hello guys. Today I want to talk about D3.2 inheritance for IB biology. Mission didn't teach this and we just did the Kognity, so I thought it would be good if I share the what things I think is important for this unit. And in this video I will talk about the 9 concepts I think is important and also the 5 skills that I think will be very useful for this unit. So let's get started. Okay, so the first thing we want to understand is haploid versus diploid. And this is supposed to be very easy because it's we go through this in mitosis and stuff. But remember, haploid is half, so you only have 23 chromosomes, and in diploid you will have 46. And what's different is you will find haploids in these gametes. And these are the sex cells, which is the eggs and the sperm. And then when two of this haploid come together, the 23 plus 23 is 46, right? So they will end up with 23 pairs of chromosomes. And you will find it when the gametes, the egg and the sperm meet to form a zygote. So everything about inheritance is about how these two haploids can form one diploid. So, oh, I should also add, we label haploid as N and then diploid as 2N. So that's a notation thing, but this is important to know as a basis for inheritance. Okay, so we understand the haploid versus diploid now. Now we should understand the homozygous versus heterozygous. So homozygous versus heterozygous. So homo means the same, and zygous, we're talking about a zygote here. So we're talking about some diploids versus heterozygous. And hetero is different, right? Heterosexual is a prefix. So basically, when we are looking at a trait, we have a pair of chromosomes, right? And then the specific part where it codes for the trait is over here, let's say. And then this is allele. Allele. So when we want to say homozygous, it means that the allele for this pair is the same. So for example, we will notate it as like AA if it's the big ones, if it's dominant, which we'll get to later, or it could be both small ones, TT. It's just an example. So the two letters need to be the complete same. Now for heterozygous, it means that the two alleles are going to be different. So maybe one is this and one is this, right? And so we will have A or like TT. So one dominant, one recessive, or like other configurations. Other things that we want to know from this concept includes this thing. It's called a carrier. So a carrier is when this particular gene displays it and has this has a recessive one, but it doesn't show. It's not in the phenotype. And we'll get into that in the next section, the next concept. Okay, so genotype versus phenotype. This is a short one, but just remember genotype is the actual genes. So it will say AA, or let's use T actually. TT, or let's say TT or TT. It's the 3 possibilities, right? And the phenotype is what it actually shows. So in a case that the big T is dominant, this will show um the dominant trait, right? This will show the recessive trait because there's no dominant. And because both of this is dominant, then it will show the dominant trait. So that's the difference between a genotype and a phenotype. So let's count 2 concepts in one. Okay, so now we need to talk about, let me erase it, dominant versus recessive. So dominant versus recessive. So these are both the alleles, right? We're talking about alleles here and recessive. But you can see down here in another concept, let me just check this first. We also have incomplete dominance and codominance, so I think I'll talk about it together. So dominant alleles is when this allele has the effect on a phenotype. The phenotype is the trait that it will display. For example, the color, or if it has fur, maybe other traits. Allele has an effect. and it is always expressed when it is present. Expressed when present. Now, recessive is more special. So it doesn't have an effect if there's a dominant one, but if both are recessive, then it will have. So it only has an effect on the phenotype, which is the trait it displays, Just good to say it again. Only when no dominant ones. And it's actually not just like black and white here. There's also things that will happen, including codominant ones, right? Net alleles. So this is actually where both pairs of allele will affect the phenotype. And I think the best example I always like to use is, let's say one allele codes for the white color and one allele codes for the red color, and both are dominant. So what happens when both of these alleles are present is you end up with this pink color, right? We all learn the color theory, right? We all learn the color theory when we are younger, so this is how it will happen. And the dominant and recessive will be in charge for the how the traits are inherited down by the parents, because one of the allele will come from the father and one from the mother. Okay, so let's see what I want to talk about next. Phenotypic plus why is it so thick? Phenotypic plasticity. So this is a concept. Okay, so uh, that's not the right one. Need to be there should be like erase all button, I think. Phenotypic plasticity, whatever. Plus, how to spell it? Plasticity. So this is it's what we say it when there is a organism can display a different phenotype depending on the environment. So they're adapting kind of different phenotype, which is the trait. So you will see a different thing depending on the environment, right? And I think there are some examples we're supposed to know here. Uh, but I'll just talk about the concept right here. So this is advantageous for the organism to do because um, when their environment is changing, like they can adapt. For example, when it is like winter, they can make their skin white to camouflage when it's snowing. I'm not very sure of a specific example, but some are permanent changes. Permanent. So imagine like you get a tattoo and you can't take it off. That's basically what happened. So it also doesn't have to be like a thing that you can see. It can be a physiological or behavior change, right? So basically, it's an interesting thing where behavioral change. So it's a way to adapt, and it also impacts natural selection because the phenotype is displayed in a genotype, right? And then so if this placidity is advantageous to their survival, then it will be more likely to pass down. So the animals we see today have the most advantageous phenotypes depending on their environment. Okay, so now we'll get into one of the harder parts, which is the blood type. And this is, you need to use up your brain a little bit more. So not all of the traits are coded with only two different alleles. For sometimes it is three. B. So let me just do a Punnett grid. Right. So these are the three different alleles that cover blood, and um, I shouldn't have used all blue, but it's okay. I hope it's okay. Sorry. Um, but yeah. So for example, if you have the A type and A type, you end up with A type, and these two are the dominant ones. These two are dominant. So for example, when A meets B, we will get a new blood type called AB. So let's fill this in. This is recessive, so when it meets a dominant one, these two are dominant, remember? We will get this A blood type, and just fill in A and B goes to AB, B and B has just B, B and the recessive I, you just stay with B and just fill in it like A, B, and this will have an O blood type. And why do we need to know this is because um it we need to know about it because of our blood transfusions, right? Because sometimes you need to donate blood, but actually these different blood type they have serious health consequences. So um the A blood type, it has the A antigen. Okay, so now I will talk about the most confusing part, in my opinion, about the cognity, because I don't think it write about it very well. So we have some sex-linked disorder. So this is the issue that has to do with the X and Y of the sex-linked chromosome, right? And the specific example that we need to know is called hemophilia, and it is an X recessive. So what this means is that, for females it is X and for males is XY, right? Well, because it's recessive, so let's say the always define your this. It's helpful. This is normal, but the small h is bad, right? So for the females, they can have, and this is only tied to the X. So this is X of big H and X of H. Because this is a sex link, that's what it means. So when you have two X's, you will either have like X, H, X, 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 X. So for females is only this, because it is a recessive, only this will have the hemophilia, right? And this will be a carrier because it don't philia. This will be a carrier because it has this problematic gene, but it won't display it because it's recessive, and this is all good, normal. But for males, if only one of their if only one of their things have a problem, if only one of their X's have a problem, and we already know that X comes from the mother, so we will talk about it in this pedigree chart too, right? So if only one of them has a problem, then there's only one X, so there's no dominant X to cover it, so they will have the hemophilia. So the males cannot be the carriers. Write it right. This is a small h. So hemophilia and normal. And the same thing goes for the dominant or Y sex link, so diseases. And there are some examples in the textbook that is cool to know. But this is a very abstract idea, but I will give you guys the examples when I'm talking about the cognitive practice question that I missed. All right, so now we're looking at cognitive practice questions, and I want to share the ones I got wrong. I did very bad, but I would like to explain why I got them wrong. Well, not, but like why there is the right answer. So this question asks, if a male with hemophilia and an unaffected non-carrier female had a child, what is the probability that the child would be an affected male? So for this one, I would recommend to draw out the Punnett grid. So the small h is the always label this. This is a good habit. If the small h has the allele for hemophilia and the big H is unaffected, and we know for hemophilia it is a recessive thing, so the small h need to appear twice for all the h's for it to take effect. So the male with hemophilia, we know that he has hemophilia, so the only X gene has a small h, and of course a Y, and an unaffected non-carrier means that both are healthy. So once we draw the Punnett grid and match up all the results, we see that there is 0% chance that it will be a male, which is XY, and also that the X will have a small h because this X gene is supplied by the mother, which is a non-carrier and have both good hemophilia. Okay, question 4. Okay, that's stupid. Question 6. If three genes control one characteristics, what is the possible genotypes produced? So it's asking for genotypes, not the genotypes, not the phenotype, right? So for this we need to use the equation. I don't think I talk about when I'm using my iPad. It's too late to get it now. My parents are sleeping. I want to go to sleep too. But this is a formula you want to use. If there is the n amount of genes, like here, it's 3 genes, one characteristic. So yeah, you use this formula to 3, and the correct answer is 27. I didn't know when I was doing it. Okay, question 7. So this is a pedigree chart, and here are the things that I want to share when looking at this. So it will mostly have the label, but mostly the circles are the female and the squares are the males. And we want to see like how this tracks, right? So we want to prove that this Achu syndrome is not X-linked dominant. So if it is X-linked dominant, what will happen? So we see that there's an affected male, so his X gene already have a problem. And if it's dominant, then whoever gets this X will 100% get it. So all of the females in this is F2, the filial thing too, will have to be diagnosed. But we know that because it is a dominant problem and the mother is healthy, then the X gene it will supply to a son will be healthy. So we see that there's an affected male in F1, and that proves that it is not X-linked dominant because if it was X-linked dominant, then the son should not be able to get this because the mother is healthy. So we know for sure that the mother didn't give him the problematic X, and the father give the Y, which don't have any problems. So the fact that the son from the second generation, this, if you can see my mouse here, that he has proves that it is not X-linked dominant. Yeah, so for this one, you want to look at the initial parents and you want to be able to identify like what possible genotypes they have. It's important you do genotypes and then you compare it to the phenotypes that occur. So that's how you will read a pedigree chart and identify. Question 9 and 10. So when we're calculating ratios, I have this bad habit that I never even like draw it out before. I just try to guess it. I think of it about it. Autosomal recessive disorder. So I don't think I talked about this, but let's see what Google has to say. Yeah, sickle cell disease. Oh, that was what it was about. But yeah. So if the father has a disease, then he has 2 copies of disease. So it is a recessive one. But if the mother is a carrier, she only has 1 copy. So the explanation is pretty good here. I'll let you read it yourself. And lastly, question 10. What? Okay. So if you cannot make the deductions about the sex link things and the pedigree by yourself, maybe it is helpful to memorize it, although it's pretty painful. But X-linked diseases are more prevalent in males because they only have one X, and that is the reason that they have it more because they don't have another X to potentially cover the thing that their X have a problem with, if that makes sense to you. Yeah, females have 2 X's, so their other X can override the faulty one, but males are more prevalent with X-linked disease. And if it is Y-linked, of course, no female will be affected because there's no Y in females. All right, that's it for today's video. I forgot to talk about the other skills, but Punnett score is pretty easy. The box and whisker is more of like a statistics thing. The ratio is when you just count the different options in a Punnett square, and we went over the practice questions. So hopefully you guys all learned something today, and I'll see you in the next video when maybe I talk about the HL part. Thank you.